Okay, for the second part, we are going to be having a look at genetic engineering. Now, this is a higher tier topic that we're going to have a look at. Okay, so what we need to know is that scientists can actually take out a gene from a totally different organism and then insert that section of DNA into another organism. And that is known as genetic engineering. Now, that organism that has been altered is known as a genetically modified organism. So, for example, some jellyfish glow from the dark. If you're able to take out that glow from the dark gene from a jellyfish and put it into bacteria, the bacteria should be able to glow in the dark. Pause the video and copy those notes underlining keywords, please. Okay, well done. Now, what I'd like you to do, using a whole page in your book, I want you to create this table exactly as I have done here. So if this is your whole page, this is my whole page, and I'm going to go through different bits that you are going to copy in that to explain genetic engineering. Pause the video, and when you're ready to start, you know what to do. Okay, so in step one, if you think about a scissors, in biology, the scissors in this example is known as restriction enzymes, and they cut that section of DNA that's needed with a few unbased pairs at the end. So you can see over here, these ones at the end don't have any pairs, and we call these ends here sticky ends. They then take, it says, a section of DNA containing the gene for making insulin, for example, is cut from a human chromosome in this way. So if I'm trying to make insulin, this is a way of doing it. I will get from a human cell, I will extract that section of the DNA, that gene that makes insulin. Pause the video, and I'd like you to copy the explanation in one row and the diagram in the second row. Okay, so for part number 2, it says here restriction enzymes again are used to cut the plasmids open. Now remember, the plasmids are found inside the bacteria. So I've started off with a human cell, and now I'm going into a bacteria where circular DNA is known as plasmids. And using the restriction enzyme, I cut it open. And it says there, by using the same restriction enzyme as was used in the human chromosome DNA, the cut plasmids have the same sticky ends. So these letters over here are not random at all. And the blue bit, you might want to use different colors, the blue bit shows the section that is being cut. Once you've copied that down, you can move to number 3. Right, so it says here, sections of DNA containing the insulin gene are mixed with the cut plasmids. The complementary bases, so remember the A with the T, the C with the G, the AT and the CG, on the sticky ends pair up, and then another enzyme called ligase is used to stick it together. So there's 2 types of enzymes involved in this process. And here you can see it all being stuck together on the diagram. Pause the video and copy that down. Okay, and in the final step, those plasmids are then inserted back into the bacteria. You can see where that gene is. And we know that bacteria, they divide so quickly. So that means in no time, there'd be a lot of insulin produced by these bacteria that can then be extracted and used to create insulin injections for diabetics. So this is an excellent example of genetic modification used brilliantly in the medical field. Pause the video and copy the notes and picture. Okay, you need to know what are the pros and cons of genetic modification. Of course, we know we can grow crops with desirable characteristics if we perhaps take from another type of plant. We might have more fruit or more resistant to disease. Okay, so that might mean that you have more food for less money. This is particularly important in developing countries. Okay, so for example, beta carotene is used to protect people from growing blind. So being able to put a gene into a plant that allows it to produce beta carotene would be incredibly helpful. The con is that sometimes you don't know how genetically modified plants may affect our health. So there's not a lot of certainty about this, but it has caused people to be concerned. The other concern is that it could potentially change the ecosystem. Also, in the wrong hands, it could create enormous ethical issues. Pause the video and make some notes on the pros and cons of genetic modification. Okay, let's see if you can apply what you learned. So here's an exam question. Describe how the wild cow has been selectively bred to produce large volumes of milk. Give yourself 5 minutes to answer this and then check your answer. Right, and here we have answers. I would put very specifically what the characteristic is in the answer. Okay, I want you to go back to that question I asked you at the beginning. What are the key differences between selective breeding and genetic modification? Speak to your neighbor, see what you can come up with, and that's excellent work. Don't forget to go to Show My Homework to see what homework has been set. Well done. After this, you need to go and update your revision cards at the back of your book if there is time.